So with that, it's my great pleasure to introduce today's keynote speaker, Nick Goeden, co-founder of and vice president of technology at Capsida Biotherapeutics. Prior to co-founding Capsida, Nick led a team developing the novel adeno-associated virus, AAV engineering technology that underlies Capsida's biologically driven gene therapy platform. During his tenure as a postdoctoral fellow at Caltech, he developed high-throughput methods for screening combinatorial libraries to explore the AAV fitness landscape and engineered novel AAVs with high efficiency and specificity for the rodent and primate brain. So with that, I'll hand it over to you, Nick.
Hi, everyone. It's wonderful to be here today, and I mean that in every sense of the word. Having grown up in Los Angeles, I never thought we would have a Biotech summit in Los Angeles. I never thought for the last year we'd actually have an in-person event again, and certainly I never thought that I'd be speaking at this, so it's wonderful to be here. As Kathy mentioned, my background is in bioengineering. I did my undergraduate work at Caltech many years ago at this point. Went over to USC for a PhD in developmental neuroscience, and then back to Caltech for a postdoc in Viviana Gradinaro's lab, where myself and my co-founder developed this technology and spun out a company. I'm actually not going to start with Capsida. I will touch on that briefly at the end, but since today is all about Los Angeles and biotech, I figured I'd start with a little bit of history of my perception of biotech in Los Angeles. For me, your choices in biotech for many years were Amgen or not Amgen. That was really it. And there's nothing against Amgen. Amgen's wonderful, but a lot of us have been wondering why is there not a bigger biotech scene in Los Angeles? We have top-tier institutions. I myself have degrees from USC and Caltech, which is nothing against UCLA or their football team, but we have great institutions. We have large pharma partners here. What's been missing for a lot of years are organizations like this that can bring these resources together and top-tier venture funding. So our company exists because Westlake Village Biopartners started about 3 years ago. They've raised almost $1 billion in funding for biotech, primarily in this area, and they're one of the partners that funded our company. You can't start companies in LA without people to make space to put things in. Alexandria Real Estate is that company that we have lived in. We've moved 3 times, but they've always been ARE buildings. They do a phenomenal job. So the infrastructure is coming for LA to be a new biotech hub, which as a native is phenomenal to see. Now for me, I didn't put it on the slide, but this has always been the great funnel of depression when it comes to drug development. You start with 10,000 compounds for a target. You want to develop something for osteoporosis, and if you're a PhD, you go to the lab and you do an ELISA for 5 years, and you're involved in that first funneling process of going from 10,000 molecules down to a few hundred. You spend a few more years working in preclinical models. Maybe you write regulatory documents for a few years, which I'm sure is an experience that's worth doing a few times, not for a few years. And after 10 to 15 years of work, you have a compound. And it's an incredible journey, but it's a journey. I was talking to somebody out on the patio this morning who said the life cycle of their companies has been about 10 years. It's 10 years of your life to start a company. Well, it's always been 10 years of your life to develop a product, and that's a long time. I think COVID has done a lot of things to all of us as people and to this industry, certainly. I was talking to an executive at Lilly who told me that his 28-year career at Eli Lilly, in that career, they went from 5 years to develop a drug from concept and get it into people. And over that 28 years, that 5-year development cycle went down to 3. And that was his crowning achievement, was 5 years to 3 years. And then COVID hit, and they developed a therapy in 3 months. And he was like, I can't go back to 3 years. They formed 3 teams. They worked shifts around the clock, 3 different teams, 24/7, to develop an antibody therapy for COVID. And after 3 months of round-the-clock work, their first dose went on the private jet and flew to New Jersey and went into Chris Christie. It was just a wild story. I think there's a lot of stories like that throughout this pandemic. I think scientists as a whole sort of straightened their spine and were like, this is what I've been training for. And I think that's a lot of us don't work in the vaccine field, certainly. Even if our relatives at Thanksgiving like to think we do, we don't necessarily work on COVID vaccines. But I think it's changed the way we look at drug development. And ultimately, that person I was talking about decided I can't go back to a 3-year life cycle, and he left and he joined us to lead our drug development programs. And if you look at the coronavirus vaccine tracker today, that's not a funnel of 10,000 molecules to one. Like, there's a lot of companies who spooled up efforts, and many of them are going into the clinic and into people today in a very short period of time. And that's incredibly exciting. So, a brief transition into adeno-associated virus, which is what we work on. AAVs are a small DNA virus. They package about 4.8 kbs. They're incredibly exciting right now when it comes to gene therapy. If you look at the figure on the right, you can see the exponential rise in publications for AAVs used for gene therapy applications, particularly in the CNS. And when you look at clinical trials that also utilize this technology, you see a delayed but a similar exponential increase in the use of AAVs for clinical applications. I think that's primarily because new technologies are being developed every day, every year for CNS applications. Whether it's research applications, drug development applications, preclinical applications, the toolbox for CNS gene therapy is better than it's ever been. I think most of us who've been around for a long time remember, I'm not going to pick on Amgen all day, I swear, but remember Amgen funding, building, and cutting entire neuroscience discovery divisions in the past 20 years, and that's because biology's hard. The brain is hard. We evolved a barrier to keep things from getting into the brain, and trying to develop therapies that get across that barrier and have good target engagement and can actually move endpoints in the clinic is really hard. But I think the tools exist today to do that, and we see this resurgence in interest in CNS therapy, especially gene therapy, because those tools exist today, and they haven't before. So I'm going to briefly go over some of those tools. Viviana's lab at Caltech, anyone looking for a postdoc, by the way, phenomenal lab. Viviana's lab at Caltech has developed a number of these tools. It's essentially the toolbox we then went and started a company around. Optogenetics, obviously incredibly exciting and impactful on the field, lets us dive in and dissect, sub-dissect circuits in the brain, how they impact disease when you fix them, how they help disease. Very impactful technology. Imaging tools, of course, have helped us understand disease better and image target engagement as we develop potential therapies. Disease models are more accurate than they've ever been before. I say more accurate, not necessarily fully accurate, but we have a better ability today to understand disease, to model disease, and to make new therapies for that disease. The last one, again, from my perspective, the most important part is getting things into the brain. So for us, that was the technology that Nick and I, my co-founder, developed and started Capsida Biotherapeutics. We engineer the delivery vehicle itself to express genes throughout the CNS. So at its core, it's protein engineering, but it's applied to an organism as opposed to a protein, and you're engineering the virus for its ability to get across biological barriers, transduce cells with specificity, and express a protein or a gene in that cell. And I'll get back to that towards the end. But looking at this life cycle of understanding disease to modeling disease to developing new therapies for disease, a lot of the tools coming out of Viviana's lab that we've incorporated into this company have really enabled us to do things that we've never been able to do as an industry before. All right, so the cool science part, my favorite part. I don't usually get to present science anymore. It's one of the things you don't realize when you start a company. You get further from the science. So it's a lot of fun to be here and actually show some of this work for the first time in 18 months. It's all been about Gantt charts, and yeah, everyone loves Gantt charts. All right, so imaging technologies first. One of the things Viviana's lab developed was the ability to not just section tissue and look at sections of tissue, but clear entire organs or entire organisms and look at expression throughout different targets in that organ or organism. So for us, as we looked to build out new viral delivery tools to the brain, being able to take the entire brain or an entire organism with a brain and to look at expression throughout that organ was incredibly useful for us as a screening tool, not just as a validation tool. Let's see if the video plays. All right. This is a really nice example of why that's useful. If you have an antibody, and here I am not picking on Amgen again, if you have an antibody in your Amgen and you want to see if it engages targets in the bone, can you section a bone? Not easily. How do you look and see if it's binding your target in the bone? How do you see if it's inducing proliferation of your target population in the bone? And ultimately, how do you take that drug forward into development in the bone and move into the clinic? Her lab developed this technique for clearing the entire bone and looking at expression of that antibody throughout the bone and seeing whether SOX9 cells proliferated when that antibody was delivered to the bone. It was very strange to see a neuroscience lab clearing bones, but that was one of the exciting things about her lab is just the sheer number of techniques that were developed there and perfected there. So in this fun video, you get to image all the way through the bone. You get to see the expression of the antibody. You get to see SOX9 positive cells proliferating throughout the bone. And ultimately, that compound was taken forward and moved into a clinical trial. Similarly, our ability to understand how neuronal circuits in the brain in real time function is better than it's ever been before. So this is live 2-photon imaging deep in the brain. You're looking at the effect of reward in a living mouse. As it gets its sugar water, you see this huge, wonderful live firing of the circuits involved in that reward. This is not obviously very related to clinical applications, but understanding disease is very difficult in the brain. And this is one of the tools that was developed that we utilize to help understand CNS disorders and ultimately lead to therapeutic development. All right, into the capsid engineering phase. So adeno-associated viruses, unfortunately, did not evolve to be useful. They evolved to infect people, primates, mice, whatever, and replicate. They are incredibly good at taking a gene and expressing it in a cell. If you want to use them for clinical applications or research applications in a specific manner, they're not incredibly useful as they exist in the wild. So this was work that was started in Viviana's lab by Ben Deverman, developed a novel engineering platform for taking viruses, inserting diversity into their genome and ultimately into their capsid structure, and then screening in vivo for their ability to cross the blood-brain barrier and express throughout the CNS. And what you can see here towards the left are novel capsids that were developed and expressed in the peripheral nervous system. So you can see the green descending up into the brain from the peripheral nerves. And as you move from the starting point, which is, I don't actually know if I have a laser pointer, but let's see. I don't think I do. I'm gonna describe. You look at AAV9, that's the starting point. It's really inefficient at expressing genes in the brain. That is a 10-fold higher dose than the capsids engineered off of it. And you look at the capsids to the right, PHBB, PHBEB, which were engineered for their ability to cross the blood-brain barrier, can deliver genes throughout the CNS much more efficiently. This raised the bar, honestly, for what's possible. If you went back 10 years and said to me, you could engineer a virus so that it gets across the BBB and expresses throughout the brain, I would have never believed that. Like, it didn't make sense. We specifically evolved to prevent that. And there's 7 different mechanisms between the virus and the expression that you need to affect to make that possible. And that's the beauty of protein engineering, is you have no idea why this works. You're just throwing spaghetti at the wall and seeing what sticks. And this raised the bar for what's possible. It honestly took longer to understand why this virus is capable of doing this than it did to make the virus itself. And that's a challenging research problem, but for clinical applications, we don't care why it does what it does. We just care that it can do something like this. Unfortunately, this virus, engineered in a mouse, worked in a mouse. It did not work in primates or in people. That long 3-year project of figuring out why this does what it does was all about figuring out why it didn't work in primates, and that's because it utilized a receptor that doesn't have a human homolog to get into the brain. It was very unfortunate for the companies that licensed this technology, and Nick and I started to think about how do we build a platform that isn't going to rely on engineering in mice. How do we build a platform that can utilize human tissues and primate tissues so that you don't have this translatability problem? Obviously, the research applications of this variant were apparent. They expressed genes throughout the peripheral nervous system, the enteric nervous system, the central nervous system. They could do really cool things like sub-dissect expression within the brain and do sparse labeling and tracing throughout the CNS. And you can make really fun videos on Amaris if you clear the whole brain, and you can do tracing studies, and it's a wonderful research tool. The ability to model disease and show proof of concept in a disease model with these capsids is better than anything we've had as tools as neuroscientists ever. And it's incredibly exciting to be involved in basic research right now in the CNS space. That's getting viruses into the brain. Nick and I thought about where is the true ceiling? Is it getting a virus into the brain? Is it getting a virus into the brain and not somewhere else? Can you detarget the entire periphery and make viruses specific to an individual organ? Within the brain, can you sub-target different populations of cells? Can you make a virus that's specific to neurons or astrocytes or microglia? Subpopulations of neurons can you express only in dopaminergic neurons and really start to sub-dissect, both for research applications and therapeutic applications, expression in the CNS? Ultimately, the answer was yes, which was exciting for us. Looking at the evolution of these capsids from left to right, AAV9 is the starting point, is mostly liver tropic. It's really bad at getting into the brain. PHP-B and EB were the second generation that enabled expression in the brain, but you still had expression throughout the periphery, particularly in the liver. And the third generation was specific to the brain. I mean, that in one figure just blew my mind, that you could take a virus and you could flip its tropism completely on its head. And within the brain, it was specific to neurons. And if you can make a virus that typically infects hepatocytes in the liver, and you can make it deliver a gene to neurons in the brain after an IV injection, you can make a virus specific for anything. If you can do that, it's a delivery vehicle that's perfect for every gene therapy application. Some more examples of cell type specificity. So we developed capsids that were specific to GABAergic neurons in the forebrain, Purkinje cells in the cerebellum, serotonergic, dopaminergic neurons. They've been used now as research tools in disease models for about 5 years. We are developing similar capsids directly in primates at Capsida. Similarly, you can see cell type specificity within the brain depicted very clearly in this proof of concept. Different payloads with different promoters injected into the same animal with 3 different colors. You see one color in neurons, another in astrocytes, and another in oligodendrocytes. So the ability to sub-dissect populations in the brain is a very powerful tool for research applications. Into some of the proof of concepts. All right, so PHP-B, you can get into the brain. In this particular experiment, we packaged a chemogenetic actuator, which I'll explain. It's like optogenetics, so instead of light activating activity, it's a drug to activate activity. It's a synthetic protein that you express on the membrane of neurons. When you administer the ligand, it makes the neuron fire. So in this proof of concept, we have an IV-delivered virus expressing this actuator in dopaminergic neurons. Wait 3 weeks, you have expression, and inject a second IV dose of the ligand. So two IV administrations, and you can specifically get the dopaminergic neurons in the midbrain to fire. And when you look at the mice on the bottom, the top one gets a saline injection, the bottom one gets the ligand, and that guy just motors. And that's because his dopaminergic neurons are firing like crazy, he's getting a reward, and he is just motoring around that open maze. You actually know it's working because the second you administer the ligand, they start jumping around the cage. It's quite rewarding to see. It's quite rewarding for them as well. It's a silly proof of concept, but it's very obvious. And as you start thinking about sub-dissecting and modeling disease with different circuits, you can start to visualize how this tool is very useful for research applications. That being said, not useful for therapeutic applications because it only worked in mice. So we ended up starting a company with a new platform that can engineer capsids like this for applications in primates and humans. I will say, starting a company in a pandemic is a unique experience. You certainly learn a lot. You certainly start with a culture and it evolves somewhere else as you switch to Zoom happy hours every Friday. It's not the same, even if we try to tell everyone it is the same. It's very difficult to start a company in the middle of a pandemic, but it's also a really rewarding experience. So brief introduction to our company. We are a fully integrated AAV engineering and gene therapy company. That means we have discovery biology, preclinical research, clinical operations, GMP manufacturing, and we are advancing a pipeline of about 10 programs in the CNS. So, a little bit about who we are. COVID also makes it difficult to document when you're doing fun things. And it never works to tell your employees to document fun. All right, so we started in 2019. This is the day we closed funding in a tiny, dingy office at Caltech. A few months later, we opened up our first space in Pasadena. We ended 2019 with 6 people in R&D, and we had a lot of fun in 2019. We could still do things. In March of 2020, of course, everyone's life changed, and we had some fun trying to figure out how to celebrate milestones as a company in the middle of a pandemic. There's a few people from Capsida here today that you can talk to and see how successful we were at that. In 2020, we moved to Thousand Oaks, where we had different challenges. This was one of our employees stringing power cords throughout the lab to get to e-power because the fires had knocked out power to the lab. New challenges, of course. Late 2020, we broke ground on a GMP manufacturing facility. So about a year ago today, and this week, we started our first engineering run in that facility, which is a very fast turnaround for a GMP manufacturing facility. Some of our construction photos. It's a really fun process to see that building stand up in a year. End of 2020, we finished with 12 people in R&D. In April, we closed a strategic partnership with AbbVie to develop therapies for 3 neurodegenerative targets. In June, we closed a partnership with CRISPR, going after Friedreich's ataxia and ALS. In July of this year, our facility opened for GMP manufacturing. And today, we're at about 100 people across the company. So it's been a lot of growth. We went into the pandemic with 12 total, and we're here today at 100. I see people in the hallway I've never met personally before, still every day. It's a lot of fun. We have about 30 open positions today. We have a 65,000 square foot R&D and manufacturing facility in Thousand Oaks down the street from Amgen. We have a pipeline of about 10 programs starting in the CNS and advancing to non-CNS TAs. And like I said, we're hiring about 30 people in R&D. So if you're interested in gene therapy, if you're interested in protein engineering or capsid engineering, you can reach out to us. We purposefully don't have a website with information on it, but I'm sure you can find a little bit about what we do and reach out if you're interested. All right, last piece of data is our data. Proof of concept expression throughout the primate CNS. So we actually have a pipeline that works for expressing in the CNS in a primates, and now we can start to utilize all those really fun research tools to develop actual therapies. All right, so thank you everyone. I'd specifically like to thank my co-founders, Nick Flitzanis and Viviana Gradinaru. Westlake Village Biopartners and Versant Ventures are the 2 venture capital groups that funded our Series A. We could not have done this without them, certainly not without their funding, definitely not without the effort they've put into helping us build this company. I want to thank Caltech. I've been there twice in my life. It's a wonderful place to do science. City of Thousand Oaks has been incredibly helpful, going from green sites to operations in a year for a GMP manufacturing facility was not something I thought we could actually do, but they partnered with us in that build and they've been tremendously helpful. Alexandria Real Estate, obviously also very helpful for getting that facility up and running. And everybody from Capsida, certainly none of this would have happened without them. And that's it, thank you.